Good afternoon, everyone.
Good afternoon.
All right, let's give this a try here. So if I do this, do the slides move?
Yes.
Good. First of all, I think it's completely appropriate that we congratulate Nana on his good news. Nana's MCAT scores came back and they were above what he needed for admission to the Liberty School of Osteopathic Medicine, who is they are very interested in him, but told him his MCAT needed to be at least a 500 or above. And what was your score, Nana?
04.
504. So 504 is above 500. So congratulations.
Congrats.
Thanks.
I think we'll have to maybe take Nana out to dinner or something like that. We'll have to decide when we do that and where we go. So congratulations all around. Tomorrow, provided weather permits, we will run our gels again, make sure we have DNA there before we start doing anything with it, and then go from there. All right. Let's pray. Father, thank you for this icy day and for this class. Thank you for Nana's good news. We celebrate that and what it means for his future. We pray now that we would use this time wisely. Thank you for all you do for us in Jesus' name. Amen. So we saw last time that the photosynthetic machinery of both bacteria that are photosynthetic, this case here would be the blue-green bacteria, and also green plants have more than just chlorophyll. They have these antenna pigments that fill in the holes for chlorophyll. So chlorophyll absorbs at these two extremes, the blue extreme, a little bit less than the red extreme. These accessory pigments essentially cover the holes. And so even though the plant and the bacteria, they can use all the visible spectrum. They have preferred spectrum. We saw that example here, where if you break the light into a prism and you ask which part of the plant makes the most oxygen, it's the parts exposed to both the red and the blue. They're still making oxygen with the other wavelengths, but they prefer the red and the blue. So that means plants have preferred wavelengths, but they can use virtually all other wavelengths. That means if you want plants to grow inside, you need to make sure they have a full spectrum light with plenty of blue light in it that will allow them to grow. Regular fluorescent lights are simply not bright enough for plants. So if we look at the photosynthetic machinery within the thylakoid membrane in the chloroplast of plants, we see that they are organized in the membrane in a very regular looking array. They're kind of arranged in concentric circles. Your book gives the idea that they're kind of in this very structured sort of array, and they are. So you have concentric circles of these various types of chlorophylls and various types of accessory pigments. These chlorophylls are bound to various proteins, and the structure of the thing looks a lot like this. So this is an LHC, a light harvesting complex. The light harvesting complexes will absorb light at the different frequencies. Because they're arranged in the membrane in a semi-crystalline fashion, they will get excited. The electrons go up to that higher energy level. When the electrons go down, that photon of light is emitted, but that photon of light then excites another LHC bound pigment. and it gets excited. And then its electrons drop back down to ground state. And then that photon gets released, but then that excites yet another LHC until you get to the reaction center. So this transfer of the photons of light, that is called exciton transfer. And I think I have it in the next, that's called exciton transfer. Exciton transfer is possible because these pigments are in the semi-crystalline array. You have this extremely efficient transfer of light energy from one pigment to another. These pigments are almost arranged in a solid state fashion within the membrane. That solid state fashion allows for this very highly efficient transfer of photons from one LHC to another until they get to the reaction center. So it is almost as though the LHC pigments play hot potato with the photon until it gets to the reaction center. Well, what do we have at the reaction center? At the reaction center, the electron goes into the higher energy level and then it leaves. The reason it leaves is that you have other molecules that can accept the electron. And the chlorophyll, which normally is a really lousy reducing agent, is converted from a lousy reducing agent to a pretty good reducing agent. And it's able to reduce the next thing in the reaction center until it gets to the bottom. When it gets to the bottom, that particular molecule will accept that electron and become negatively charged. Well, the chlorophyll up here has lost an electron. It is positively charged, but that's okay because this charge is separated across the membrane. Charge separation across the membrane stabilizes it. Therefore, the transfer of this electron is in fact quite fast and quite effective. And several laboratories have been able to use microbeam lasers that are very, very fast. And they've discovered that the speed at which this electron is transferred is in the picosecond range. It's very, very fast. So the reaction center takes that quantum of energy from the LHCs, that photon of light, and they are able to take the reaction center chlorophyll, excite it. It now goes from being a lousy reducing agent to being a really good one. And it will be used to give electrons to an electron transport chain that will give those electrons to NADP to make NADPH. So the reaction center with all of its LHCs, its antenna pigments, its accessory pigments, that constitutes what's known as a photosystem. So this picture right here, all this stuff here essentially. All that, and this is just half of it, that constitutes a photosystem. PS, photosystem. So the electron passes from the accessory pigment. That electron goes back down. The photon gets transferred to the reaction center chlorophyll. It goes up, and now the electron is going to leave. And it will leave and go to an electron acceptor right here. So the chlorophyll, reaction center chlorophyll, will reduce this electron acceptor. This electron acceptor is on the same side as the membrane as chlorophyll. So now that electron acceptor gets reduced. It's now negatively charged. And this here is meant to be the reaction center chlorophyll. So you can see that reaction center chlorophyll. It goes to the excited state. This electron goes up here like this. There it is. This electron then leaves and goes there. And you have charge separation now. Except that charge separation is going to get even better because this electron donor is going to give its electrons here to the chlorophyll and re-reduce it. That charge will now be here. And remember, there's a membrane on either side here, so that charge gets absolutely separated across the membrane. So the first reaction centers that were examined in great structural detail were the ones in bacteria. And that was a good choice because they're much simpler in bacteria than they are in green plants. So the crystal structure of the first crystal structure of a reaction center was the crystal structure for Rhodopseudomonas. And let's see, it was Rhodopseudomonas sphericum? No. I forget which species of Rhodopseudomonas it was, but the Rhodopseudomonas is quite simple when it comes to its reaction center. Now, the purple bacteria, to give you an idea of the types of photosynthetic bacteria, the two main ones, well, the three main ones are the purple bacteria, the green bacteria, and the blue-green bacteria. And if we use our makeshift blackboard here, we can go to our camera. So of the photosynthetic bacteria, so the main groups of photosynthetic bacteria, there's purple, the green bacteria, and the blue-green. So all the work done on the blue-green bacteria says this is a natural phyletic, monophyletic group. That's pretty well established. The purple bacteria come in the purple sulfur bacteria and the purple non-sulfur bacteria. The purple non-sulfur bacteria have been further split. With the purple sulfur bacteria, that seems to be a pretty natural group. This group is anaerobic. They're obligate anaerobes. So they do business without any oxygen. Now remember, this particular group of bacteria, they will split water to make molecular oxygen. These do not use water. These use hydrogen sulfide. So these will end up producing, so they'll take sulfide, they'll either oxidize sulfide to elemental sulfur, or they may oxidize it further to sulfate. The purple non-sulfur bacteria are among one of the most remarkably diverse groups of bacteria when it comes to their metabolism. So where these are strict phototrophs, they're photoautotrophs. So these will take Carbon dioxide, they use something like under the Calvin cycle to assimilate carbon dioxide into their cellular carbon. Many of the purple non-sulfur bacteria, they are photosynthetic, but they can also utilize organic carbon. And many of these can exist either in the presence or absence of oxygen. So this group is metabolically extremely diverse. Very complicated group, very interesting group. The green bacteria also have the sulfur, and you guessed it, the non-sulfur green bacteria. And it's kind of the same thing, except it's pretty clear that the green and the purple bacteria are not related. They're distinct. So once again, these are obligate photoautotrophs. PAT, photoautotrophs. These are also obligate anaerobes. These guys, once again, they're very diverse metabolically. These guys can either work with or without oxygen. They can do photosynthetic, so they can be photoautotrophs, but they can also be heterotrophs. So these are your three groups, and these have phycobilisomes. That's a feature of the blue-green bacteria. It's also a feature of the red algae. The red algae also have phycobilisomes. And are thought to be among the most primitive of the algae. Green bacteria have something in their cells. They're these little tiny vesicle-like structures underneath the membrane where their photosynthetic machinery is. These are called chlorobium vessels. Chlorobium vesicles. Those are specific to the green bacteria. In fact, there's a green sulfur bacterium whose genus name is Chlorobium. The purple sulfur bacteria, they have in their membrane, they have all these deep folds. In their membrane, and those deep folds are the site where you have your photosynthetic machinery. Deep folds. Nothing fancy like chlorobium vesicles or phycobilisomes or something like that. Very good. Back to the ranch. So the Rhodopseudomonas is a purple sulfur bacterium. I hope that puts that in context. So its photosystem has 4 different polypeptides that hold the pigments and everything else in place. This also forms a semi-solid state arrangement in the membrane. So you get very fast exciton transfers. And here is the crystal structure of the Rhodopseudomonas virilis photoreaction. So up here, you have C-type cytochromes. Those are your C-type cytochromes. Remember, C-type cytochromes have the heme covalently attached to the polypeptide chain. You have several of them. Light is going to hit, as you can see here, there. You have a bacteria chlorophyll, and it will give its electrons to the accessory pigment, this other bacteria chlorophyll, which is over here. So you go from here to here. So this transfer goes this way, like that. Then you have something called bacteria phaeophyton. Bacteria phaeophyton has all the structures of bacteria chlorophyll, but it does not have the magnesium in the center. And that's over here. So we go from the bacteria chlorophyll here to over here. And then the bacteria phaeophyton gives its electrons to the quinone. The quinones are down here. So these go from here to over here. And there's an iron right here, and that iron is going to help stabilize those quinones once they take up the electrons and hydrogen ions and become quinols. The oxidized bacteria chlorophyll is going to receive electrons here, and those electrons will come through here and will re-reduce the bacteria chlorophyll. Because bacteria chlorophyll, once it gives up those electrons, it becomes negatively charged, doesn't it? The electrons that come in to save the day, they come from the hemes. Look at how fast the transfer is here. The transfer here is 3 picoseconds. So this transfer here from the bacteria chlorophyll to the accessory pigment bacteria chlorophyll, 3 picoseconds. This transfer to the quinone is 200 picoseconds. These transfers are very, very fast. That's due to the fact that these elements are inserted in the membrane in a semi-solid state fashion. So the purple bacteria, the green bacteria too, do not have chlorophyll. They have bacteria chlorophyll. They also have phaeophyton. That is one of these bacteria. Well, phaeophyton is a chlorophyll molecule that lacks the central magnesium ion. Bacteria phaeophyton is a bacteria chlorophyll molecule without the central magnesium in the tetrapyrrole ring. And purple bacteria take that reaction center bacteria chlorophyll. When it gets excited by excitons, it goes from being a really terrible reducing agent to being one that's pretty good. However, it is not a good enough reducing agent to reduce NAD. So this electron goes to an electron transport chain that includes phaeophyton and the quinone, and then to a cytochrome BC complex, to a C-type cytochrome back to the reaction center chlorophyll. In doing so, it creates a proton gradient, and this particular photosystem is used to make ATP. So this is called cyclic photophosphorylation because the electron that's ejected from the reaction center bacteria chlorophyll comes back to it. Cyclic photophosphorylation is used to make a proton gradient. The proton gradient is used to drive ATP synthesis via an ATP synthase. It is not used to make reductant. How, pray tell, then do these cells make reductant? They make reductant through reverse electron transport. reverse electron transport. Now, if we look at the photosystem of the green bacteria, we can see that this particular photosystem starts at a more positive electronegative potential than does that of the purple bacteria. So the green bacteria photosystem is excited by transferred photons, those excitons. It then becomes a very good reducing agent, so good, it's able to give its electrons to a ferredoxin, and the ferredoxin can give its electrons to NAD. This is non-cyclic photophosphorylation. This photosystem now has to be re-reduced. Now notice this system can give the electron to a quinone, just like what you have over here. That quinone can be used to drive the cyclic photophosphorylation, or it can be used to drive non-cyclic photophosphorylation, where it makes reductant in the form of NADH. This cyclic photophosphorylation is used to make ATP. However, the green sulfur bacteria, it can do one or the other. This one, the purple sulfur bacteria, can just do the one. It can only make ATP via cyclic photophosphorylation because its particular photosystem, bacteria chlorophyll, can't become positive enough to reduce NADH. The green sulfur bacteria photosystem, bacteria chlorophyll, can, and it can make reductant. So as we said, in cyclic photophosphorylation, the electrons get removed from the chlorophyll of bacteria chlorophyll. They go around electron transport chain back to the bacteria chlorophyll or chlorophyll. That movement of electrons through the electron transport chain pumps protons. You pump protons into the intermembrane space for a bacterium. You pump protons into the thylakoid membrane lumen. Those protons are then removed from that area using the ATP synthase. That will synthesize ATP. That is cyclic photophosphorylation. And the photosystem that you see in purple sulfur bacteria and other types of purple bacteria, purple non-sulfur bacteria, they look a lot like the type 2 photosystem found in blue-green bacteria and in green plants. The green bacteria, both the green sulfur and green non-sulfur bacteria, they have a different photosystem that becomes much more positive when it is excited by an exciton. It is therefore a good enough reducing agent to reduce nucleotides like NAD or NADP to make NADH or NADPH. They can, however, give that electron to a quinone, which will give the electron back to the photosystem. If that electron is given to reductant, then the photosystem, bacteria chlorophyll or chlorophyll, is reduced by water in the case of green plants, water in the case of blue-green bacteria, and hydrogen sulfide in the case of purple sulfur bacteria and green sulfur bacteria. So this particular photosystem looks a lot like a type 1 photosystem found in green plants and blue-green bacteria. And it can make reductant or it can make ATP. If it makes reductant, that is called non-cyclic photophosphorylation. Okay. And because the delta E of the type 2 reaction center pigments is more positive than those of type 1, it can be used to make reductant. You can reduce NAD to NADH. You can reduce NADP to NADPH. And so that exciton transfer of the type 1 photosystem pigments produces a molecule that can reduce NAD to NAD. It can make reductant, NADH, NADPH, or it can be used to make ATP. Now, blue-green bacteria, cyanobacteria, and green plants have the two photosystems that work in tandem. So the type 2 photosystem, like the kind that you see in blue-green bacteria, like the kind that you see in purple sulfur bacteria, is the first photosystem. And the second photosystem is the type 1 photosystem, like the kind you see in green sulfur bacteria. Reductant from the type 1 photosystem is used to reduce the type 2 photosystem. Sorry, backwards. Electrons from the type 2 photosystem are used to reduce the oxidized type 1 photosystem. And that arrangement is called a Z scheme. You find the Z scheme in blue-green bacteria and green plants, and here's how it works. So here is your type 2 photosystem, chlorophyll or bacteria chlorophyll, depending upon if you're a green plant or blue-green bacteria. Except blue-green bacteria have chlorophyll. This gets excited by an exciton. It becomes a relatively good reducing agent. It then gives its electrons to pheophytin, series of plastoquinones. Plastoquinones are very similar to ubiquinones structurally in mammals, but they are slightly different structurally, so they're called plastoquinone. Then they go to a B-type cytochrome complex that gives its electrons to a plastocyanin. Plastocyanin is a copper-containing protein that looks a lot like cytochrome C. Plastocyanin ferries those electrons to the photosystem 1, oxidized photosystem pigment. When excited by light, this particular photosystem then becomes oxidized, gives its electrons to an acceptor electron, then usually some sort of phyloquinone, acceptor chlorophyll, or pheophytin, some sort of phyllocyanin, iron sulfur protein, ferredoxin, NADP, oxidoreductase, and then you reduce NAD to NADPH. So these work in tandem, light activating both the different photosystems, converting them from relatively poor, well, not relatively, they are, poor reducing agents to relatively good reducing agents. One of them, namely photosystem 1, becomes a positive enough reducing agent to be able to reduce NAD or NADP to NADH or NADPH. The other photosystem, that of photosystem 2, becomes a good enough reducing agent to reduce the oxidized photosystem 1. So the two of these working in tandem, this is called the Z scheme because it looks like a Z that's been put on its side, so it looks more like an N. And this is a scheme you have in blue-green bacteria and in green plants. It looks like it's the combination of the photosystem from purple sulfur bacteria and the photosystem from green bacteria. And here is the photosystem arrangement for photosystem 2 from the cyanobacterium Synechococcus elongatus. So here is your photosystem chlorophyll, and here are your photosystem pheophytins. So you'll remember, you give those electrons first to pheophytins and then to plastoquinones. Here are your plastoquinones. They're held in place by iron, very much like what you had in purple sulfur bacteria. D1 and D2, these are structural proteins that keep this whole thing together. There have been some good studies of mutants of D1 and D2 in Arabidopsis. D1 and D2 mutants will create photosystems that don't work. They're no longer in that solid state arrangement, so you can't transfer excitons very effectively. Both D1 and D2 have these tyrosines that are strategically held to be able to transfer electrons. The photosystem, once it is oxidized, is re-reduced by the oxidation of water to make protons and molecular oxygen. So the enzyme that does that is called the OED or OEE. OEC, oxygen evolving complex. The OEC has been purified. It's not a big protein, but it has this core of manganese ions. And these manganese ions go through a cyclic kind of oxidation reduction. It changes oxidation states, and that's how it's able to take water and it's able to oxidize water so that you make molecular oxygen. Those electrons go through a pathway not unlike what you see in photosystem II in purple bacteria. So once the electrons have been liberated from the reaction center pigment, that transfer the electrons through the electron transport chain, because that electron transport chain is right there, right next to the pigment, is thermodynamically spontaneous because you go from a relatively positive delta E electron carrier to a relatively more negative one. That means transfer the electrons down the electron transport chain is thermodynamically spontaneous. The membrane holds the electron transfer components of the photosystem in a solid state configuration. So transfer of electrons is quite fast, very efficient, and occurs within the picosecond range. And here's PS1 in plants. So you have your LHCs, lots and lots and lots of LHCs with their antenna pigments. You get the electrons to the reaction center chlorophyll, shown here. You excite it, you give up the electrons to nearby chlorophylls, to pheophytins, to plastoquinones, to the ferredoxin, acetoreductase, and then to reductant here on the stroma side. So you make your electrons for reductant, you make reductant on the stroma side of the thylakoid. Plastocyanin migrated from PS2 to PS1, and we can see it right up here. This is a beautiful crystal structure picture of the pigments and the polypeptides of PS1. You have this beautiful trifoil kind of structure. Take away all the proteins, you have the different pigments. Here's your iron sulfur center that's at the bottom. If we go back to this, the iron sulfur center sits essentially right across here at the bottom. See that right there. The production of oxygen re-reduces the pigment from photosystem II. It produces molecular oxygen, therefore. This particular Z scheme is called oxygenic photosynthesis. And the electrons pass from photosystem 1 from the ferredoxin to the B-type cytochrome. This particular B-type cytochrome is unique, so it's called an FB6F. F stands for frond. It can return those electrons back to photosystem 1. That's cyclic photophosphorylation. It'll make ATP. But if not, it'll give those electrons to photosystem 1. And that's how the Z scheme works. Now you have a problem if you have a Z scheme. To show you that problem, let's go back to the Z scheme slide and I'll show you what I mean. Can you see that any sort of photon that excites this photosystem can also excite this one? And if that's the case, then your photons might end up, instead of properly activating this one, then activating this one, they might all end up acting over here. Or if you have photons that are meant to excite this one, they can also excite this one. Because these are the wavelengths at which these are excited overlaps, you can get essentially all the photons going to photosystem 1 and most of your energy being made to make reductant. Why is that a problem? Let's take a look why that's a problem. So because less energy is required to excite photosystem 1 than 2, excitation of photosystem 2 could generate excitons that instead of activating photosystem 2, they immediately migrate over to photosystem 1. And that would leave photosystem 2, instead of being reoxidized, it would leave it in the lurch. And that would prevent it from generating enough high energy electrons to properly reduce photosystem 1. So if photosystem 1 gets over activated, it will not get re-reduced. So this is called exciton larceny. Exciton larceny. And exciton larceny is more of a problem if you have lots and lots of light in the middle of the day, when you have lots of blue light essentially hitting the plant, hitting the leaf, hitting the whatever. So to prevent this problem, plants will essentially short circuit the Z scheme. They will spatially separate photosystem 1 and 2 in the thylakoid membrane to make this work. So consequently, photosystem 2 is organized in the tightly opposed membrane stacks of the thylakoid grana. So they're in the granal lamellae. PS1 is found in the unstacked membranes of the thylakoid, that is the stromal lamellae, what the old electron microscopists called frets. They're in the frets. And here's a picture from your book, essentially, that shows this. So here is the opposed part of the thylakoid. Here are the stromal lamellae. Notice the stromal lamellae have a lot of photosystem I, and it's not really associated very much with photosystem II. Here in the opposed, the oppressed granulamellae, you have more photosystem II, light harvesting photosystem II. And it gets associated to a large degree with photosystem I. So here's two. Two's in the granule stacks. One tends to be more out here. Now notice that the light harvesting complex for photosystem II, it tends to mediate the apposition of the membranes in the thylakoid. That's not by accident. The light harvesting complex for photosystem II has an extension that inserts into the membrane of the opposite thylakoid vesicle. By inserting into that opposing vesicle membrane, it mediates apposition of those stacks. The ability of the light harvesting complex of PS2 to do that is regulated by light intensity. So in bright sunlight that has a high blue light component, photosystem II absorbs more light than photosystem I. You get a big pool of reduced plastocyanin, much more plastocyanin than PS2, PS1 can use. And that big pool of reduced plastoquinone activates a protein kinase, and it phosphorylates the extramembrane threonines. Those phosphorylated threonines are now quite polar. So now that extramembrane piece of the PS2 light harvesting complex cannot mediate the apposition of the thylakoid membranes. So essentially they look like this here. They look like a dead whoopee, so to speak. As we're here, they look like a live whoopee, and they're able to insert into the membrane and mediate the proper apposition of the membranes. And the association of the light harvesting complex of PS2 with PS2 and then also with PS1 in some cases too. So in the presence of lots of light, you activate that protein kinase because you have large pools of reduced plastocyanin. And now the light harvesting complex cannot mediate apposition of the thylakoid membranes. In dimmer light, as those pools of plastocyanin are used up, now that protein kinase is not as active, the protein phosphatase will predominate. You dephosphorylate the threonines. Now the light harvesting complex of PS2 can mediate apposition of the membranes again. So by phosphorylating that little piece of the light harvesting protein complex protein component, you reduce the ability to properly oppose the thylakoid membranes in place. That causes dissociation of the light harvesting complex from PS2, and now it moves to the stromal lamellae where it associates with PS1. By increasing the activity of PS1, you accelerate the oxidation of the reduced plastoquinone pools. And in less intense light with a greater red component, so say later in the day, photosystem 1 oxidizes the plastoquinone faster than PS2 can generate it. That increase in oxidized plastoquinone activates the protein phosphatase that dephosphorylates the light harvesting complex of PS2. Now it mediates apposition of the thylakoid membranes, and you have more stacked thylakoids. So electrons from PS2 will temporarily get stored in plastoquinone. Those electrons will be given to cytochrome B6F, that complex, and then to the soluble copper-containing plastocyanin, which will carry the electrons to PS1. The components of the cytochrome B6F complex have a high potential and a low potential B-type cytochrome, not unlike complex 3 in our own cells. And this complex even undergoes a Q cycle, just like the Q cycle in complex 3 of our electron transport chain. Here's a crystal structure of that cytochrome B6F complex. And because the hemes take the electrons from the plastoquinone one at a time, those electrons get cycled back to the plastoquinones and then back to the hemes, and then the electrons go one at a time to plastocyanin. So the plastoquinones keep getting loaded up with electrons. That pumps more protons. That's the Q-type cycle. Very much like what you see in in complex 3 of mitochondria. The OEC, an old name for the OEC is the water splitting complex, or WSC. This complex will take electrons from water and use it to re-reduce the pigment, the oxidized pigment in photosystem 2. So this complex, the oxygen evolving complex, has a 4 manganese array. Tom Kunzelman did his PhD on this 4 manganese array. So electrons from the water get passed to a tyrosine residue of the D1 subunit. And then they will go to from the tyrosine to the pigment. Tyrosine is an aromatic residue. It can tolerate some extra electrons for a brief period of time. Because this is in a solid state array, the transfer of electrons from the OEC to the D1 tyrosine to the pigment is very fast. So the manganese complex goes from completely neutral oxidation state to a +1, to a +2, to a +3, to a +4, and then back to 0 again. So it seems to withdraw electrons from water to the tyrosine. It holds the oxidized water in place, releasing protons into the lumen of the thylakoid, greatly increasing the number of protons available for proton pumping, and eventually releasing molecular oxygen. The exciton energy is the energy that drives this reaction from here to here. Those electrons released from the photosystem pigment, central reaction center photosystem pigment, go through an electron transport chain. It can generate reductant, but it can also generate a proton gradient. Just like what we saw in animal mitochondria. Those electron carriers alternate in the electron transport chains, alternate between those that carry both protons and electrons and those that carry just electrons. They also go from the most positive delta E to the most negative delta E, making the transfer of electrons down the chain thermodynamically spontaneous. Protons get extruded into the thylakoid lumen. That creates a stark pH difference between the thylakoid lumen and the stroma. Because the stroma is being deprived of protons, it becomes relatively alkaline. pH inside the lumen of the thylakoid, however, becomes rather acidic. Therefore, you have a remarkable capacity for work. And as those protons get pumped back into the stroma, they can make lots of ATP, and they do just that. So you not only have a disparity in concentration, you have a disparity in charge and a disparity in pH within a completely closed membrane. Your proton motive force, therefore, in this case, is formidable. Formidable. So we go from PS2. Those electrons may go to PS1 to re-reduce it. Those electrons can actually be given back to PS2. You can pump protons just like what you show here, or they can be used to make reductant. Either way, you're going to make some increased, you're going to make a proton gradient, an acidic gradient in here. Those protons will be returned to the stroma via an ATP synthase. The structure of ATP synthases in plants resemble greatly those in animals, and those genes seem to be orthologous. And this ATP synthesis via photosynthesis, via light-mediated electron transport is called photophosphorylation. Now, as we said, the ATP synthase molecules, they work the same and they look pretty much the same. So in the mitochondria, you're pumping from the matrix into the intermembrane space back into the matrix, and you're making ATP on the matrix side. In bacteria, you're doing the same thing, pumping protons into the intermembrane space back into the cytosol, making ATP on the cytosolic side. In bacteria, in chloroplasts, that gets a little bit flipped because of the nature of the chloroplast. Now from the stroma, so you've taken this and turned it upside down. You go from the stroma into the thylakoid lumen, out into the stroma again, you make ATP on the stromal side. And the stoichiometry has been worked out pretty well. In the case of plants, the proton concentration inside the thylakoid lumen is very low. You have a high proton concentration, very low pH. And it seems as though you make 3 ATP for every oxygen molecule that you produce. That's different than what you see in animal cells. And that is due to the fact that you're pumping protons into a closed area, closed vesicle. All right, we are out of time. So we will take this and go to the bank. So we'll pick this up on Friday. We will have a quiz on Friday, weather permitting. And I'll see you guys in lab tomorrow at 1 o'clock. And again, let's try to make sure that we find some time to celebrate Nana's good news, because it is very good news we're celebrating. And so I will get back to you with that, and have a great rest of your day.
You too.
Thank you. For the quiz on Friday, is it just should we set our questions on photosynthesis?
Pretty much, that's what we've covered. So photosynthesis, yeah.
Okay. All right. Sounds good. Thank you.